Everybody, I'm not going to talk a lot about E&L in this particular talk. This is about other aspects of qualifying single-use systems, which some folks in the audience here may not be familiar with. So I had a lot of preeminent speakers before me all day speaking about E&L. I will highlight the kind of synergies with E&L that we consider during qualification of various single-use items, okay? So first, I'll talk about design considerations. We'll look at fit, form, function assessments, which are important, especially when you're trying to dual source or multi-source materials to fit in existing space. I'll talk about prototype testing, and I might mention some unexpected things that we've noticed during prototype testing in the past. Talk about the general compliance requirements, which you've been hearing about all day. And then I'll talk about the physical qualification of the materials. How do we introduce them, and what supporting activities happen around that introduction? And finally, I'll just mention life cycle management. Extractables and leachables is not a slide of its own, so we'll move on. So everybody is familiar with the manufacture of biopharmaceuticals, which is what we're talking about within the J&J world, at least at the moment. And there are many different types of biopharmaceutical, from MABs or multi-specific antibodies, therapeutic proteins, antibody drug conjugates, vaccines, cell and gene therapy products, and orphan drugs, okay? And they're used for all kinds of therapeutic areas, prophylactic for preventing disease, oncology, neurology, autoimmune diseases, and so on. So pretty much every therapeutic area you can think about, okay? So we have a typical biologics manufacturing process shown on the slide. So we start with some vials of frozen working cells from the working cell bank. These are thawed and grown up in various technologies. You can use orbital shakers, roller bottles, generally of increasing numbers of cells to inoculate the next manufacturing stage throughout the manufacturing process until you get to the production bioreactor step, okay? Where is the only place you really want to start producing the actual product molecule. And in fact, you may inhibit the production of that molecule at earlier stages of the process. Then the product is harvested somehow by, say, centrifugation or depth filtration, followed by sterilizing grade filters into a harvest storage vessel. And then the purification steps begin with normally some kind of protein A chromatography stage. The next stage, quite typically, would be something like virus inactivation, followed by polishing chromatography steps, typically CEX and AEX, cation and anion exchange respectively, or some other type of chromatography like hydrophobic interaction chromatography or mixed mode anion exchange. After that, the second orthogonal virus removal step happens, which is like a nanofiltration step, and the last stages of manufacturing then of the bulk drug substance at least are that UFDF step, which has been mentioned I think in quite a few presentations today, and then the formulation and the freeze of the bulk drug substance in a large scale biotainer prior to shipping perhaps to another site for drug product fill, which is shown in the last picture there on the slide. In between each of these stages, you obviously have pooling vessels for collecting and storing the product in between each unit operation, okay? The key point is you can have a manufacturing process that is entirely disposable, flow paths, everything from the vial thaw step through to the bulk fill and the drug product fill. and all of the, I suppose, the challenges that come with that throughout. So what are the design considerations for single-use systems? Well, they're surprisingly similar to reusable systems. Are you building a new facility, or are you fitting into an existing facility, right? You have to design your equipment and processes around that. What's the nature of the specific unit operation that you're looking at? Is it cell culture? Is it downstream, fill finish, and so on? Okay. What's the operating environment that the equipment has to sit in and work in? Is it a fully qualified clean room, grade A, grade B, or is it in a non-qualified area, right? So that will determine how you can connect and make transfers to and from the equipment that you're qualifying or designing. What's the operating time, temperature, pressure, and so on, right, that your equipment needs to run at? Is it adequately sized? What are the safety concerns for your particular process? Is there anything specifically required for, say, not contaminating a future batch of product with something that's very, very potent that you can't necessarily show through cleaning validation that you can remove, right? And of course, then the associated hardware, you need access for the maintenance and cleaning and calibration and so on for that. And then the other more, I suppose, esoteric things, interfaces with the monitoring and control systems for your manufacturing process. And any other connections you need to other equipment or utilities, okay? Other considerations, how do you sterilize or sanitize the material that you're using? Are you buying in irradiated components, which we've spoken a lot about throughout today? Are you going to autoclave it yourself or steam it in or out of place prior to use? So that's obviously a very key concern. And of course, there's more and more people don't want to be doing assembling in their manufacturing facilities. So we want to bring stuff in that's components in that are ready to use, okay? So irradiated components is probably the most common, right? Unless we're going to steam a filter in place or put it in an assembly and autoclave it and use it elsewhere. The compatibility of the plastic material in the single-use system with the process fluids you're going to expose it to needs to be considered, okay? However, in general, our biologics manufacturing processes are pretty benign in terms of not having very notable incompatibility issues with some of these commonly used plastic materials. And then we have to consider the extractables and leachables, which we've spoken about a lot today. So they get a star note on each slide where I mentioned them, right? And what's the impact of the extractables and leachables, not just to patient safety, but to the processes as well? Are they going to hinder cell growth, for example, in a mammalian cell process? And I think most people have seen cell growth issues due to BSD-TBPP, which has also made a star appearance on a number of talks during the day. Or can you have, for example, issues with antibody drug conjugates caused due to a leachable species interfering with say an assay, or actually really damaging the activity of a particular antibody drug conjugate? And finally, then, I suppose robustness is something to consider. Is it going to be easy to get another source of a single-use material in the event of a shortage, right? So we've all gone through COVID. We've all seen the challenges to keep manufacturing medicines when particular components were in short supply. So fit, form, function assessments become important in an existing facility when you want to multi-source, okay? Can it fit where it's needed to be placed, right? Does it have to connect to existing equipment? is it dimensionally suitable? So we ran into an issue with a filter, and it wasn't even a product contact filter. This was for buffers or media, and we needed to fit it into a particular pipe run on the facility. And we actually couldn't get one that would fit into the space, right, that we had to install that. And we ended up having to do a different workaround. Okay, we were not able to dual source that filter. The form, right, the compatibility of the single-use material with the process fluids, and vice versa, right, that the alternate material isn't going to damage your product. And again, that would be maybe particular concern around filters and filter validation, where we go to a lot of trouble to show the product doesn't damage the filter membrane, or the filter membrane doesn't damage the product. Again, we've got the E&L concerns that we've mentioned already. And then we can sometimes make the form work if the form is different, right? But it probably means you have to buy new hardware, which will delay changing out or multi-sourcing a component. Okay. And again, the new hardware probably won't have the same geometry and shape as the current hardware, so you might have some extra work to do around qualifying that. All right. And then finally, does it do the same function, the same job? And does it do it satisfactorily, right? And there we have seen, I suppose, concerns over things that were nominally the same, just very simple piece of tubing that was supposed to have the same durometer rating from two different suppliers. However, one, we couldn't get it not to kink when we were trying to use it in the space we had to use it. Okay, so we weren't able to proceed with that particular dual sourcing. And then, do you have to steam it on? Can you steam through? Right, those are big concerns, right? So, for example, if you set up to steam on components onto a bioreactor, a steam through connector is a major engineering change to actually install it. Because you're going to have to install steam traps, extra drains, new automation. So those are the concerns with the functionality and performance of the equipment. And can you sample in the way that you're used to sampling, right, from a bioreactor or another component, a single-use mixer? And those factors all need to be carefully considered when you're multi-sourcing. And how do you document that? Where is that documented, right? So there should be a formal documentation system. These were all the factors that were evaluated, and we have deemed this is suitable to implement. The project, if you're dual sourcing or indeed designing a new facility, you should get prototypes in and test them so that you avoid those nasty surprises that I alluded to earlier. Don't skip that bit, and you will be under pressure because we want to shorten the deadline for delivering the project always, right? There's always the next product coming down the line that's putting you under pressure to try and get finished early. But it's very important to do that prototype testing, and it allows you to identify those issues that you may not have identified in a paper-based fit form function assessment. Again, another example using filters is one filter, the current filter could filter a solution faster than a proposed alternative. And that may or may not matter, right? So it depends, okay? In this case, it did matter because we're trying to increase the number of batches per year going through the facility. But 12 months ago, maybe it would not have mattered at all. Okay? You can identify unexpected issues, like I've mentioned already, and there could be a little bit of reiteration of the design and reiteration of the prototype testing. The compliance requirements, and again, not to exclude any of the ones that have been mentioned already, the forthcoming chapters from USP or the Chinese Pharmacopoeia, but we would like ideally BSE, TSE, animal derivative free materials, or compliance with EMA 4101. We would like very clear statements that the materials are not derived from human materials. And clearly that's low risk for a lot of SUSs. We would like clear statements that the materials haven't been exposed to an environment that has been used to manufacture any highly sensitizing or toxic agents, as per the list there, right? And if they are, how is the SUS controlled to make sure that there's no risk to our customers or patients? Okay. Melamine contamination, again, probably low risk for most polymers, but again, just a clear statement stating that there is little or no melamine risk here, or negligible melamine risk. And the standard biocompatibility, USP 87 and USP 88 and or ISO 10993. And I guess, right, we're going to see less and less USP 88. We thought that it would only be for new components. However, some suppliers are actually removing it from certificates for components they've already certified as being USP 88 compliant. And that's a bit of a challenge for us internally to deal with. pH EUR 3.1.9 for silicone elastomers and tubing. Again, probably more relevant to the bulk drug substance and drug product world, but we would like to see it. Somebody else mentioned it earlier on today, I think it was before lunch, for the drug substance manufacturing as well. And statements on sterility or control of irradiation or gamma rays coming in. And also, what's the recommended shelf life and storage conditions and transport conditions? And again, they're not often very special for any of our polymeric materials, but on occasion we kind of get a few oddballs that do come our way. Don't store below 0 degrees, or don't expose to temperatures above 30 degrees, which may or may not be a challenge in each individual case. So then, when we're introducing any new single-use material, whether it's existing dual-sourced material or a brand new material coming in from a new vendor, we must meet those compliance requirements that we mentioned and the forthcoming requirements as well. The manufacturing environment at the SUS manufacturer must be suitable, okay? And this would be covered partly with a vendor audit and a questionnaire that gets filled in, where we look at what's the grade of manufacturing area, ISO 8 or better, for example, for manufacturing single-use components, and what other controls are in place for particulates and bioburden and endotoxin for the single-use components. There would have to be a quality agreement in place with the supplier, and then they have to be made available within our ERP system or SAP. And then we would have, the internal specifications to allow us to release the materials when they're received at the user sites. And then there are all the supporting activities that happen at the user sites, right? So we'd have mixing validation to support mixing in single-use mixers or single-use bioreactors. And there may be a requirement to do some dissolution studies for solutions that are difficult to manufacture, which quite often applies to certain media solutions, can be difficult to make, okay? We would have to generate chemical hold study data for buffers and media in their storage containers. Quite often you'd be looking at things like pH and conductivity for buffers. For media, it would be the performance of the media in actual cell culture operations based on a particular hold time for that media. For bioreactors, we'll obviously be looking at their ability to control the metabolites of the cells at the appropriate levels throughout the cell culture process during PPQ. and the ability of the system to maintain its pH and so on throughout the manufacturing process, and even, you know, to avoid foaming, for example, by anti-foam addition. Okay. Of course, for all of the unit operations, microbial integrity is very important. And for cell culture processes, we will carry out media challenges, which maybe just follow the manufacturing process to make sure we're in control of the axenic boundary throughout the manufacturing. The other thing that we might look at for bioreactors would be KLA studies to show that we can transfer oxygen into the cells at an appropriate rate, okay, which will feed in a bit to the sparging strategy, and then there has to be a feeding strategy developed for the bioreactors per product. For filters, we look at bioburden control or bacterial retention, depending on whether it's at the end of the manufacturing process. So anytime before a long-term storage, we would generate bacterial retention data to support the filter use in operation. And then for intermediate hold steps or buffer holds or media holds, we'll do microbial sampling of the buffers and media and processing sums to show we're in control there. And then there are various CSV and continuous process verification activities that will happen supporting all of the manufacturing. And then if you're using a hybrid system where some of it is reusable and some of it is single use, obviously there's cleaning validation around those reusable skids, particularly around things like UFDF or centrifuges where you're not using disposable centrifuge technology. Integrity and leak rate testing can be important, again, with filters as well. We talked about we talked about E&L risk earlier, and the identification of those SUSs based on a risk assessment, which was just presented in the last talk by Isabel, and the capability of the various unit operations to clear potential leachable species from the manufacturing operations, okay? And particularly chromatography, where you've got a bind and elute step and you're eluting into a brand new matrix, gives you a very good opportunity. Or that difiltration process where you're changing out the process volume 8 to 12 times, shall we say, during manufacturing activity. Then the safety assessment performed for higher risk materials as per the Bioforum and USP guidance that we spoke about already. And if we're exceeding any of the ICH guidance values, we need to consult our toxicologists to make sure that the material is safe to use for a given commercial product. And the other point then is just to note the potential for leachables to interfere with cell growth or other aspects of the product or process. Finally, we get to change management. So we need to manage the single-use systems throughout their entire working life in our processes. And of course, we have two types of change. We have the ones that the users want to do to the single-use components to annoy the suppliers. And then we have the ones the suppliers do to annoy the users, right? So, and there's been a lot of that, I guess, in the last period of time in both directions. And then maintain that compliance of the single-use system throughout the life cycle of the product, okay? Not forgetting the E&L risk that may arise from any change throughout that life cycle. So in conclusion, we talked through, I suppose, the design and qualification considerations for single-use systems. It's multifaceted and does involve a lot of different disciplines, okay? And we looked at, some of the other concerns, how are we sanitizing or sterilizing the single-use system before we use it? And a key point is the compatibility of the single-use materials of construction with the process fluids And through our process characterization, we can show that the single-use system isn't damaging our products. And ENL is in red there again, just to highlight it. And the robustness then factor is quite important. It can be surprisingly difficult to get an alternative component from another vendor to fit into that ecosystem that you've bought into when you designed and built your facility day one. Okay, so that's it. Thank you for your time and patience.